So this how Robert Hooke discovered what he calls cell, and we also know years later, we know of Anthony Balian Hooke, who was the first to observe a living cell, a microorganism specifically, under a simple microscope. We have in 1838 and 1839, Matthias and Theodore, that discover the cells in plants and animals, ended up the conclusion all animals and plant cell, all animals and plants are made up of cells. We also have a discovery in 1855 by Rudolph, who stated living cells come from other living cells, classical of mitosis, cell division. So this information put together give rise to what we call a cell theory. And we know what cell theory is. Cell theory, there are theory forwards, explaining those findings that are happening over a lengthy period of time. What are those theories? The first one says all living things are made up of cells. Cells, more or less, so any living thing you see made up of a cell. Either it being a single cell or multicellular organism. We know we can remember organization of life where you have some cells existing as a single cells. You know, some cells existing as filaments, pyrogyra. Existing in form of a colony, while multicellular like a more complex organism, me and you. Aisha.
Sir?
Please. Interesting. The second one says cells are the smallest working unit of all living cells. So the smallest, just from the definition, the smallest functional unit of a living is a cell. Anything less than a cell may not be considered living, even though we know of the theory of endosymbiote, some organelles that historically were known to exist independent of a cell. What are those organelles? Can somebody tell me what organelles are these organelles?
Yes, sir.
Which organelles are these ones?
We have nucleus.
What? Go ahead.
We have nucleus, we have Golgi body, we have ribosome, lysosomes, and
I'm talking about the endosymbiote. Historically, there are some of those organelles that were thought to exist from history, were thought to exist on their own before they now live together with cells over a longer period of time. Now they become more of independent. But initially they are dependent, they are on their own. They are independent. Now, because they exist over a longer period of time, they become independent. We have a chloroplast, we have mitochondria. So mitochondria and chloroplast were initially considered to live on their own. That is why they are considered endosymbiote. And one thing we also know, we see that these organisms, these are organelles that have their own, nucleus, they have their own DNA, genetic materials. So this is basically more of a history. The history proof that these are actually independent before they become dependent over a lengthy period of time. And then we have all cells come from pre-existing cells through cell division. And classically, what is cell division? We know how a cell gives rise to a new cell. That one we are very much familiar with. Okay. Before we move, we have to talk about the different types of cells. We have two types of cells, eukaryotic cells and the prokaryotic cells. This division, Moses.
Sir?
Can you, are you trying to say something?
Yeah, the other one is a single cell, while the other one has a double cell. So,
which one are you talking about? Which one?
The eukaryotic cells have a single cell, while the prokaryotic has a double cell.
Okay, I'm not yet there. I didn't ask you. I'm just trying to explain first before I ask the question. Okay, so can you mute yourself, please? So, these cells are divided into two. You have a eukaryotic cells and the prokaryotic cells. What is the basis of the division of these cells, eukaryote and the prokaryote? It is based on the presence of a membrane-bound nucleus. And the first one we have the prokaryote, pro, from the name, before carriers. So before this nucleus, before the membrane, the nuclear membrane, because all of them contain the genetic material. But one of them, the genetic material, is found suspended within the cells. That is in the case of prokaryote, before the formation of this membrane-bound nucleus. While in the case of eukaryote, you have a membrane-bound, or you have the DNA, the nucleic acid, the nucleic material bound or contained within an envelope called a nuclear membrane. So on the basis of DNA, which is the main basis of the classification, you have one enclosed within a membrane-bound vesicle called nucleus or nuclear membrane, while the other one, you have the genetic material suspended within the cell. Now, on the basis of size also, we have the prokaryotic cells are usually very small, whereas the eukaryotic cells tend to be bigger in size. In terms of organization, they're usually single cell. We talked about organization of life where we mentioned some cells exist as single cells, while some as filament, some as a colony, while some are multicellular. So prokaryote usually exist in form of a single cell, while eukaryote exists in most cases as multicellular cells. With respect to metabolism, you have some of these prokaryotes require no oxygen, even though, of course, there are some that also require oxygen. And there are some that when there is oxygen, they utilize it. When there is no oxygen, they utilize another thing. They can use a sulfur, they can use a nitrogen for their own metabolism. While in the case of eukaryote, we need oxygen. With respect to organelles, just like we mentioned in the case of DNA, the DNA is not enclosed within a membrane. So also you have other organelles do not have a membrane, while in eukaryote, you have organelles that were separated by membranes. We'll see why these membranes. We'll talk about cellular compartmentalization. Why do we even have membranes?
Our team need machinery space.
Now,
How you don't do the issue
Esther.
with for
Esther.
your and she the good thing about it is that
Esther.
she's good to like if you have an issue, is it a meeting?
Esther, good. Okay. So, in a human body, we have a different type of cells because overall in a human body contain about 100 trillions, about 50 to 100 trillion cells. All these cells belong to at least 200 or 210 different type of cells. These different type of cells, each has its own specialized functions. These cells come together to give a tissue and organ. They multiply and obtain several copies of themselves. But these cells majorly differ in size, in shape, and the lifespan. We can see the red blood cells. The red blood cells are very small in size. We have an ova, which is a reproductive cell, which is very big in size. With respect to shape, we know that red blood cells are concave, a bit spherical in shape. But we also know sperm cell, which is a male reproductive cells that are long. We also know nerve cells that are also long. With respect to lifespan, we know the shortest, like sperm cells that only live for only 3 days. We have red blood cells. Before red blood cells, we have test receptors on our tongues that can last for only 10 days or thereabout. We have red blood cells that can live for 3 to 4 months. But this is in exclusion of the sickle cells, because the sickle cells live a very short period of time, shorter than the normal red blood cells. We have the skin cells that live for only 1 month, and after that, they will have to die and the new ones will be produced. We have muscle cells that can stay for years, like 15 years. We have a nerve cells that can stay for a lifetime. So we have a different type of cells. From the picture here, we can see some of these cells based on the structure they differ. Some are spherical, some are normal, concave in shape, like red blood cells. Some we can see they are very long, like muscle cells, like nerve cells, like sperm cells. The pardon me with the included amoeba and paramecium. They are not supposed to be here because we are talking about cells in the human body. But we can include them when we are talking about cells in general. And you can see also the shape of amoeba. You can see the pseudopodia that enable the movement. And you can see the shape. Even so, the shape of amoeba can change depending on the activity. It produces all these pseudopodia when it is in motion. And we know that has to do with the cytoskeletons that help or facilitate the movement of the organism. And we also have paramecium with the cilia that also facilitate the movement of the organism. So this is a typical type of typical cell and animal cell, and we know an animal cell is made up of majorly three different ones. I don't know what this thing is. Okay. So cell can be divided into three main parts. And that is the cell membrane. And we know what the cell membrane is. A more or less a gate, a security gate that protects the cell, that prevents substance from the surrounding environment coming into the cells, and at the same time prevent substance from within the cells going out of the cell. Also protect the cells from any harmful substances. We also have a cytoplasm. The cytoplasm is more or less a fluid. And that fluid, or a space that contains fluid, that is found between the plasma membrane and the nuclear membrane. We contain that fluid suspended with organelles. This contains some cytoskeleton which regulate the movement of the organelles, the movement of some vesicles, the movement of substances from one place to another. Sorry. We also have a nucleus, which is the third part. And this nucleus, we know, this nucleus contains a genetic material we call DNA. Within the nucleus, we have the DNA, and we have a particular space where so many biological processes or reactions like transcription, translation, replication takes place, synthesis of ribosome, and so many other things. Okay, this is the structure of plasma membrane. I will talk about in detail the composition of plasma membrane still within in this class today. And this membrane, we said the major functions is separate the extracellular and the intracellular environment. It gives the protection to the cells. It regulates the movement of substance in and out, and also facilitate the interaction with the neighboring cells. We talked about how this plasma membrane or cell membrane facilitates the interaction with the surrounding environment and the surrounding cells around. We talked about the glycoproteins and glycolipids shortly. We also talked about cytoplasm, where we say cytoplasm is in space, a cavity between the plasma membrane and the nuclear membrane. And we say it contains this fluid. And this fluid is where the organelles were suspended. We can see the nucleus also suspended as a bigger organelle. And we can see the smaller organelles also suspended within the cytoplasm. We also have the nucleus, where we said the nucleus contains the genetic material, and within the genetic material, and in addition, we have a place we call nucleolus. And within that nucleolus is where the DNA replication, transcription, synthesis of ribosomes, and quite a number of things takes place inside. And also state that the nucleus is being separated from the cytoplasm by what we call a nuclear membrane or nuclear envelope. And this nuclear envelope separates the cell, separates the cytoplasm from the other from nuclear component or the component inside the cell. Pardon me, please. I'm sorry. Hello? I'm fine, sir.
What issues are you talking about? Your brother has something like that. Like 2 years ago that happened. What?
Sorry. So we have this genetic material all inside the cells, and we said it is enclosed within what we call a nuclear envelope or nuclear membrane, where you have a bunch of a very long DNA of up to 2 meters if it's to be stretched, but all condensed within a very small organelle that is not to more than 5 micrometer. Maybe I think there is somewhere ahead where we talked about how this genetic material being condensed or compacted inside the nucleus. So we have this nuclear envelope, and within the nuclear envelope contains some openings called nuclear pore, where substances can get into the nucleus and then out into the cytoplasm. So these are basically the 3 main parts of a cell. We said that cells can be divided into 3 major parts: the cell membrane, the cytoplasm, and the nucleus. And we talked about what are the roles of these 3 major components of the cells. We talked about them. Now the next thing is, we talked about the plasma membrane. What are the components of this plasma membrane? This plasma membrane is made up of a phospholipid bilayer. Even though, in addition to the phospholipid, there are quite a number of substances. So we have, number 1, lipids. Number 2, we have proteins. Number 3, we have carbohydrates. All come together to give cell membrane. We talked about this component one by one and how, in which form they come together to give a cell membrane. Okay. Before then, the composition of a cell membrane depends on the function of a cell. Some cells may have a higher lipid content, while some cells may have an equal lipid and protein. Some cells may have a significant amount of carbohydrate within their own membrane. So depending on the function of the cell, just like give an example of red blood cells with almost equal of 49% protein and 41% lipid. We can see cells from the liver, hepatocytes. We have about 60% protein and only 40% lipids. Like this, we have an inner mitochondrial membrane that contains about 75% protein, only 23% lipids. So the composition of this component of a membrane depends on what is the function of that cell. For example, like the myelin sheath of nerve cells that serve as an insulator. It has about 79% lipid, only 20% is protein. So This is basically the concept that the function determines the proportion of each of these components. But in all of them, of course, you will find this component, whether it's lipid, protein, and carbohydrate. All you can find in a cell membrane. But the composition of them differ on the function of the cell. Now starting with phospholipid or membrane lipid, phospholipid. What is this phospholipid? The phospholipid contain fatty acid or contain, let me start it. It contains two parts. We can see the structure there. Can we see my cursor? Class.
Yes, we can see it, sir. We can see it.
Yeah. So you look at this. You can see this, something that look like a head.
Yes, sir.
And this that look like a tail.
Yes.
So phospholipid contain a head and a tail. This head is nothing but a molecule we call glycerol. You can see the molecule glycerol here if you can see my cursor here. This is a molecule or a compound we call glycerol. So this glycerol, because it contains this hydroxyl group, therefore it is polar in nature. It can easily interact with water. Because when we say polar, means it can easily interact with water. Non-polar, something that cannot interact with water. Are we together?
Yes, sir.
While this tail,
Yes, sir.
this tail is fatty acid. So this fatty acid does not easily interact with the water. Therefore, the one that interact with water is called hydrophilic. That is why here we say hydrophilic head, meaning the head is hydrophilic in nature. Here we have hydrophobic tail, meaning the tail is hydrophobic. Hydrophobic means water hating, something that doesn't like water. Now we can see here from the structure, how does the fatty acids and the glycerol come together? Now we can see here, we can see the first one, we have the fatty acid. This is a fatty acid that contains how many carbon? Let's count together. 1, 2, 3, 4, 5, 6, 7, 8, 9. So this is a fatty acid that contains 9 carbon, right? So this 9 carbon fatty acid is now attached to this hydroxyl group of glycerol. So you can see the glycerol here. You can see it here. Can you see it?
Yes, sir.
Yes, this is the glycerol.
Yes, sir.
So the another fatty acid here, which is different from the other one. Let's count this one also. 1, 2, 3, 4, 5, 6, 7, 8, 9. This is also 9 carbon fatty acid. It's also attached to the carbon 2 of glycerol here. But the third hydroxyl group of carbon 3 is not attached to fatty acid. Rather, it is attached to a phosphate group, a phosphorus group. Moses. So it's attached to a phosphorus. We call it a phosphate group.
I'm getting
Moses.
Okay, sir.
Please, can you mute yourself? Moses.
Yes, sir.
Please mute yourself.
I'm getting you, sir.
Please, can you mute yourself? Good. So it's attached to a phosphate group. The phosphate group is also attached to another group, a different group. And this group could be so many, it could be, we call it, can be choline, can be ethanolamine, can be serine, can be inositol. So this group, all the other, it can be fatty acid, different fatty acids. Some of them, this one we counted 9 carbon fatty acids. Some are 10 carbon fatty acid, 11 carbon, 12, like this, 14, 15, 16. Some of these fatty acids, which I just didn't mention, you can see this one is straight, while this one bend. The bending here is due to a double bond here. When a fatty acid have a double bond, we call it unsaturated fatty acid. While a fatty acid that does not have a double bond, we call it a saturated fatty acid. So unsaturated fatty acid will have a double bond. Due to this double bond, you can see the bending it causes. And this bending in a lipid, if there is a phospholipid that contain the fatty acid that have this double bond, the double bond will bend to form what we call a kink bond. So this kink bond help in determining some physical properties of a plasma membrane. One of them, for example, is fluidity. Is the plasma membrane held to be too rigid or it is flexible? If you have phospholipid in the membrane that contain a lot of fatty acids that have a double bond, that are unsaturated, you can have a membrane that is very fluidy in nature, that is very flexible in nature. But if most of the fatty acids that are found in the membrane are made up of saturated, there is the chance that that membrane is going to be rigid. Are we together? So, another thing I need to mention also is the length of the fatty acid. Some fatty acids, like I said, they can be 16 carbon, it can be 14 carbon, it can be 12 carbon, or lower than that. Are we together? So the smaller the carbon, the length of the fatty acids, the higher the fluidity. The longer the chance that the membrane is going to be rigid. Is that clear? Now, we have known what is phospholipid. And the thing we need to know is that phospholipids are of different types. It could be phosphatidylcholine. This is when a choline is another molecule, is attached to the phosphate group here. It is called phosphatidylcholine. When it is ethanolamine that is attached here, it is called phosphatidylethanolamine. When serine is attached here, it is called phosphatidylserine. Likewise, inositol. In the case of sphingomyelin is also a phospholipid, but not like these ones. It is sphingolipid. It is called phosphosphingolipid. Why is this phosphosphingolipid? Simply because it does not contain glycerol. Rather, instead of glycerol, it contains another backbone we call sphingosine backbone. In addition to that, that sphingosine backbone attached to phosphate, and it does not attach to either ethanolamine, choline, serine, inositol. Rather, it is attached to a sugar. Are we together? We have a typical example of a glycolipids, which we will talk about shortly. Okay, now we've known what is a phospholipid. And one important thing we know about the phospholipid is that the phospholipid contains the hydrophobic and the hydrophilic. How does this phospholipid give rise to a cell membrane. So there is one important thing I will mention about the characteristic of a cell membrane. The cell membrane is self-healing. Self-healing in the sense that it can heal. You know what is healing, right? It can heal itself. So the explanation here I will give will tell you why it is called something that can heal itself. Another thing is fluidity. I've already explained fluidity with respect to the length of the fatty acid, with respect to the saturation level of the fatty acid, which I have one thing to mention also with regard to the presence of cholesterol, which I will do that shortly. But what is this self-healing? Now you have a phospholipid. You know what is a phospholipid? And this phospholipid, we know it contains the hydrophilic, the water-loving, and the hydrophobic, the water-hating. When you suspend or when you have this molecule and you put it inside the water, what happens? Because the water-hating doesn't like water, therefore, the water-loving will come together and will be the one to interact with the water molecule. So that the water-hating will now be on air. They doesn't want to interact with water. So classical, we obtain something that look like this. We call monolayer, where you have the hydrophobic, the hydrophilic region here, the water loving, will now come together and they interact with the water, assuming this area is the water, the offward is the water. So they will arrange themselves in this way so that only this water loving part is the one that will interact with water. While the water hating, which is the fatty acid tail, will not interact with water. That clear? When you put these things enough, and maybe assuming you just check the water, what will happen? You are trying to force them to enter inside water, that they must interact with water. Do you know what happens when you do that? When you do that, they can form what we call a bilayer. So they will arrange themselves, say, those parts that we don't like water, let's come together. They will cover themselves to form this structure we call a bilayer. So you have this part that doesn't like water, they come together, while the other part will now expose to water. Up here we have water, so they can easily interact. Down here we have a water, they can easily interact. But in between, they don't like water, so they will come together. So you will not see water here. Even when you find this phospholipid inside the water, they will interact in such a way that this area water will not enter, but this area water will enter. They can also form what we call a micelle. So they form a cycle like this. Here inside, water cannot be inside. But outside, water can interact with water. But inside cannot interact with water. Or they can also form what we call lifosome. In this case, you have a bilayer. So inside here, you can have water inside. Or anything fuller. While here, you can also have anything fuller. But in between, you cannot have anything fuller. This is what we call a lifosome. So this is exactly what happened in the case of cell membrane. The layers of these phospholipid form a bilayer in such a way that inside the cell, you have the cytoplasm, and the cytoplasm contains substances that contain water inside. So you can have water inside, while outside also is the extracellular fluid can contain water. Is that clear? While in between, you have the area that contains the fatty acid where water cannot be part of that. So this is how a cell membrane is formed.